Today I would like to explain the differences gene expression between prokaryotes and eukaryotes. Gene expression is a biological process which assembly of protein from encoding gene. It starts with the use of information from a gene to synthesize functional protein. This process occurs in prokaryotes and eukaryotes for adaptation to changes in environment. Differences of gene expression between prokaryotes and eukaryotes. These are the differences between them. It will be explained in detail later. First, prokaryotes are polycistronic whereas eukaryotes are monocistronic. In prokaryotes, mRNA is polycistronic, that messenger RNA which encodes for two or more proteins. In eukaryotes, mRNA is monocistronic, that mRNA which encodes for only one or specific protein or polypeptide. Next, prokaryotes are coupled transcription and translation whereas eukaryotes are not coupled. Prokaryotes are single-celled organisms that lack a nucleus, and their DNA floats freely in the cytoplasm. To make a protein, both process transcription and translation occur simultaneously in cytoplasm. No post-transcriptional modification occurs. In contrast, eukaryotic DNA is located inside the nucleus and then transcribed into mRNA. The newly synthesized mRNA is then transported out of the nucleus into the cytoplasm, where ribosomes translate the mRNA into protein. Besides, prokaryotes do not have post-transcriptional modification whereas eukaryotes have. Before transport to cytoplasm, mRNA will undergo post-transcriptional modifications that are adding cap at 5 end, adding tail at 3 end, and splicing, removal of intron and joining together of exon in nucleus. Finally, a mature mRNA is produced. Capping comprised of enzymatic reactions. First, RNA triphosphatase removes leading phosphate group from mRNA 5-terminal triphosphate group. Then, guanylyl-transferase added GMP to produce guanosine cap in a process called guanylylation. At last, guanine 7-methyltransferase methylate the guanosine cap, attached to 5 of transcript. Capping protects mRNA from 5 exonuclease. A polyetail is formed by adding 30 to 500 adenines to the 3 end of the RNA chain. The mechanism is governed by a series of enzymes and factors. Cleavage and polyadenylation specificity factor binds upstream of AA at 5 end. Cleavage stimulatory factor F interacts with downstream U sequence and bound with CPSF to form a loop in RNA. Cleavage factor 1 and 2 work together with CPSF and later slow polyadenylation process. Polymerase stimulates cleavage at polyacide, binds to Ad12 at 3 hydroxide group. Poly binding protein 2 accelerates addition of the by PAP signal PAP to stop polymerization after addition of 200-250. In addition, prokaryotes have single RNA polymerase whereas eukaryotes have 3. In bacteria, all transcription is performed by a single type of RNA polymerase. A single RNA polymerase synthesizes all the 3 types of RNA: mRNA, tRNA, rRNA. Whereas in eukaryotes, transcription is achieved by 3 different types of RNA polymerase: RNA polymerase 1, 2, 3. These polymerases differ in the number and type of subunits they contain, as well as the class of RNAs they transcribe. That is, RNA polymerase 1 transcribes ribosomal RNAs, RNA polymerase 2 transcribes RNAs that will become messenger RNAs, and RNA polymerase 3 transcribes small RNAs such as transfer RNAs. Thank you.